Hello, my name is Aditi Dhawan. I go to Arnold Beckman High School, and today I will be presenting on the challenges of Toxoplasma gondii and global infectious disease control. For an introduction, I am a rising senior in Southern California. Outside of school, I am involved with the school yearbook and I am a Girl Scout. I am also involved in biochemistry research and multiple medical programs as I am an aspiring physician and public health advocate. An introduction to toxoplasmosis. It is a disease caused by Toxoplasma gondii. Normally, Toxoplasma patients develop self-limiting influenza-like symptoms. It is asymptomatic in immunocompetent persons, but severe in immunocompromised persons, such as patients with cancer, HIV, AIDS, etc. These patients develop fatal encephalopathy, which is a disease that causes brain dysfunction. This leads to impacted cognitive abilities, behavior, and even consciousness. The current treatment is a combination of sulfadiazine and paramethamine. To get infected, the definitive host for these parasites are cats, as seen on the left in the image. They shed T. gondii in their feces, and humans can get the chronic infection by eating raw or undercooked meat. The acute version is transferred through tachyzoids, by drinking contaminated water or through transplantal transmission if affected during pregnancy. This means that the baby can get infected through the mother. This disease is incredibly important to global health due to the amount of cases in the world. Over 40 million people are infected by Toxoplasma gondii in the United States, and it's estimated that up to 1 in 3 people in the world are infected by Toxoplasma gondii. There is also an abnormally amount of high cases in South America, as well as high health impacts. The strains cause more severe disease. Additionally, failures in the treatment of toxoplasmosis have been reported in South America, suggesting the acquisition of resistance against current treatment methods. As you can see in this infographic, there are significantly more Toxoplasma cases in South America and even in parts of Africa. It is also important to note that no data is collected in certain areas of the world, such as much of Africa and parts of Asia. However, this is a recurring problem in global health due to geographical location as well as little funding. It is essential to collect this information so that global health workers can recognize patterns and accurately prioritize what dangers to account for. In France, raw meat and undercooked lamb consumption has led to high T. gondii rates, while in Brazil, water contamination from poor sanitization plays a bigger role. Different regions face different routes of exposure based on culture, environment, and infrastructure. What makes Toxoplasma gondii more than just a biological puzzle is how it exposes gaps in global health equity. In low-resource areas, access to clean water, proper sanitation, prenatal care, and consistent health education is limited. These are the very factors that increase exposure to T. gondii. In fact, the same disease that may go unnoticed in the U.S. can be deadly in parts of South America or sub-Saharan Africa, where healthcare systems are so overburdened and yet so underfunded. In pregnant women, a lack of early detection can result in stillbirths or lifelong complications in newborns. But it's not just about one parasite. It's about the systems that fail to protect people from these preventative diseases. As scientists, however, if we can develop drugs that are low cost, effective, and accessible, we are not just treating a disease, but we're closing the health equity gap. This is the intersection of science, medicine, and justice, and why research is so important. Much of my work in the lab remains confidential. However, I will base my information on previous literature done by this lab. A bit about toxoplasma structure. Parasites of the apicomplexa are named for their distinctly polarized cell apex that contains a number of unique organelles. These organelles are critically important for invasion and survival of host cells. They have two populations of microtubules, spindle microtubules and subpellicular microtubules. In the image, you can see the subpellicular microtubules in the apical pole ring. Microtubules have an essential role in toxoplasma. Not only does it keep it crescent-like shape, but it has an extremely important role in replicating and dividing, otherwise known as mitosis, which essentially keeps the toxoplasma thriving in the body. There are other functions of microtubules listed here on the right. So, the lab exposed the Toxoplasma to oryzolin, which is a microtubule disrupting drug at low and high concentrations. This was monitored in the lab through electron microscopy and immunofluorescence. As you can see, the oryzolin binds to alpha-tubulin in the Toxoplasma. In the results, you can see the changes in Toxoplasma structure. When treated with low-dose oryzolin, which is about 0.5 micrometers, it disrupted subfollicular microtubules. Parasites became round instead of crescent shape. Spindle microtubules, on the other hand, had remained functional, so nuclear division and budding continued. Parasites were temporarily non-invasive, but fully recovered upon drug removal, regaining shape, microtubules, and replication capacity. In contrast, when treated with high-dose oryzolin, around 2.5 micrometers, it disrupted both spindle and subfollicular microtubules. Parasites enlarged, failed to divide, and formed anucleotide zooids, cells with no nuclei. These zooids could bud and escape from the host cell but were not viable. They couldn't replicate or divide again. And so you can see on the images on the left, the image on the top has the most microtubules, but as you increase the concentration of oryzolin, you can see the microtubules slowly start to fade. Now we come to future targets and key takeaways from this research. Since oryzolin disrupts subfollicular microtubules and inhibits host invasion, this points to a druggable weakness in the parasite structure. Also, oryzolin affects T. gondii microtubules at very low concentrations, but it doesn't affect the host cells at the same dose. It suggests that we can target the parasites without harming human cells, which is crucial for drug development. In conclusion, Toxoplasma gondii is a widespread and often overlooked threat. Its transmission methods, impact on immunocompromised individuals, and global health disparities in data collection make it a key challenge in infectious disease control. By understanding its biology and targeting its unique structures like microtubules, we can move closer to safer and more effective treatments. Here are the references to my presentations as well as my acknowledgments. Thank you so much for listening to my presentation. I appreciate everyone for sticking along and hopefully gaining knowledge in an extremely important global health issue. My email and LinkedIn are linked on the screen in case anyone wants to contact. Thank you so much.